Good afternoon, everyone. My name is Syuan-You. I'm a PhD student working with Dr. Shinsuke Akehara at Gulf Coast Research and Education Center. And today I'm going to share some of my research results regarding Blackberry production in Florida. So actually, I'm supposed to present in Chrome Memorial section, because of some logistic reasons, I was pulled out from the Chrome section, and this presentation was put in the vegetable section. Okay. So berry crops are a low carbohydrate fruit. They are considered a superfood which packs major nutrients with bright flavors and intense color. Among them, blackberries are rich in phytochemicals, including anthocyanins and flavonoids, which can protect us against heart disease, cancer, and diabetes. There is a growing demand for blackberries in recent years because of the new release of varieties and improved aroma, flavor, and also the high antioxidant can bring us some health benefit. The import of fresh blackberry increased from 43 million tons in 2010 to around 74 million tons in 2017. And among berry crops, blackberry is the fourth economically important small fruit in retail sale in the United States. In the United States, major blackberry production concentrates in Oregon and California. Also, there has been a growing interest in producing blackberry in the southeastern United States. The growing area increased from 2,000 acres in 2007 to around 4,000 acres in 2017. This trend was also shown in Florida, particularly in Central Florida. However, the commercial blackberry production is still limited to local market and U-pick operations. Blackberry is a deciduous fruit crop and is well adapted in temperate climate. In Florida, its reproductive phenology is about 1 month earlier than plant grown in the temperature regions. Generally, Budbreak will occur in early March to early April, and flowers will be observed at 2 weeks after Budbreak. At about 6 weeks after flowering, fruit will be ripening in early May to June. Like in most decidual fruit crops, blackberry must be exposed to a certain amount of winter chill to break bud dormancy. The chilling requirement is highly variable among blackberry varieties, ranging from 300 hours to 900 hours below 45 degrees Fahrenheit. In subtropical climate, inadequate chilling will cause poor and erratic bud break and produce less flowers and reduce fruit yield. So for viable subtropical blackberry production, dormancy should be artificially released to increase bud break. Bioactive gibberellics, GA, such as GA3, GA4, play important roles in regulation of dormancy release in many perennial crops. The high efficacy of exogenous GA in inducing bud break has been reported in sweet cherry, peach, pistachio, and Japanese apricot. Our previous study in Central Florida also found exogenous GA3 was highly effective in advancing the onset of bud break in 3 tested varieties, but the exogenous GA3 causes flower abortion and reduced yield in a specific blackberry variety called notches. In apple, cytokinins act as an antagonist of GA during floral transition by replacing GA-associated pathways, which in turn increase flowering in apple. So the goal of this study was to optimize the application protocol of GA3 as a bud dormancy breaking method for subtropical blackberry production. We conducted a series of experiments to optimize GA3 application protocol aiming at increasing Budbreak and yield. In experiment 1, we evaluated the cultivar and GA3 interaction by applying 99 grams of GA per hectare on 3 varieties in late February. These 3 varieties were Natchez, Navajo, Wachita, and they were developed in Arkansas. And in this experiment, we found GA3 only caused flower abortion in Natchez. So, to evaluate the possible cause of flower abortions, we conducted 2 experiments. In experiment 2, we characterized the dose effect of GA3 on Natchez, where GA3 was applied at 0, 25, 99, and 198 grams per hectare. And in experiment 3, we evaluate the synergistic effect of GA3 and a cytokinin for 6-BA. The treatment included the single application of GA3 or 6-BA, or the combination of GA3 and 6-BA, and the sequential application of GA3 and 6-BA. In treatment number 5, GA3 was applied at the same timing as other treatment in late February, and then the 6-BA in treatment number 5 was applied at 9 days after the GA3 application. All the treatment were applied with a spray volume of 100 gallons per acre using a CO2 pressurized backpack sprayers in late February, during which the chill hours below 45 degrees Fahrenheit were 199 to 227 hours. As we can see, the chill accumulation was below the estimated chilling requirement of the tested Blackberry cultivars. And for Natchez, Natchez is the most popular variety grown in Florida because it has the lowest chilling requirement. All data were analyzed in product mix in SAS. In this presentation, I will only display the data of percentage Budbreak, flower number per can, and yield. And for Budbreak and flower number, we selected 5 representative flowery cans per plot before the treatment. And for each can, a section containing 20 notes was labeled and recorded to obtain the Budbreak percentage and flower number. In the first experiment, we found the magnitude of GA3-induced Budbreak varied considerably among the tested cultivars. Compared with the respective controls, exogenous GA3 increased Budbreak by 10 times in Natchez, 16 times in Navajo, and 55 times in Wachita. For the flower number per can, compared with the respective controls, flower number was increased by GA3 by 240% in Navajo, whereas exogenous GA3 had no significant effect in Natchez and Wachita. For marketable yield, compared with the respective control, exogenous GA3 increased marketable fruit yield by 560% in Navajo and by 130% in Wachita, whereas it caused a non-significant 15% yield reduction in Natchez. So in experiment 2, we characterized the dose effect of GA3 rate. At 9 days after application, Budbreak showed a linear dose response. Budbreak was increased from 24% to 50 to 59% with increasing GA3 application rate. The dose response of flower number was described by an exponential decay. Flower number per can decreased sharply from 22 in the control into 6 at 99 grams GA3, and then decreased gradually to 4 at 198 grams GA3. The dose response of marketable yield was described by an exponential decay. Marketable fruit yield decreased sharply from 2 kg per plant in the control to only 0.5 kg per plant at 99 gram GS3, and then decreased gradually to 0.3 kg per plant at 198 gram of GA. In the experiment 3, we found all the GA3 containing treatment rapidly induced Budbreak to a similar extent. At 9 days after the treatment, the GA3 containing treatment showed significantly higher percentage of Budbreak, ranging from 83% to around 87%. Then we only found 16% of Budbreak in a control. For flower number, plants start flowering at 24 days after treatment. At the end of the flowering period, at around 70 days after treatment, the GA3 containing treatment produced 87% to 92% fewer flowers than the control. The plant produced the highest marketable yield in control and in the 6BA treatment. The GS3 containing treatment had 65% to 83% lower marketable yield compared to the control. Marketable fruit yield also varied between the two treatments containing both GS3 and 6BA. The tanks mix treatment had 100.1% higher marketable fruit yield than the sequential treatment. In many temperature fruit crops, GA production is downregulated during the dormancy and upregulated during the Budbreak, whereas the exact opposite trend occurred in ABA production. So we think Budbreak induction by exogenous GA3 may be associated with antagonistic interaction between GA3 and ABA during the dormancy cycle. In addition, increased production of reactive oxygen species has been proposed to play a promotive role in dormancy release. Exogenous GA could promote Budbreak indirectly by stimulating oxidative stress responses, which have been reported in grape and Japanese apricot. And second, GA3 applied when plants are flowering can be used as a flower thinning agent for stone fruit, such as apricot, nectarine, and peach. In this study, although GA3 was applied at around 20 days before the flowering, residual GA3 level can remain above the optimal level in the tissue for an extended period, so during which flower development of notches was ongoing. And compared with the sequential application of GS3 and 6BA, the combination of GS3 and 6BA had 100.1% greater yield. Sorry for the typo on the slide. In apple, a high cytokinin and GA ratio induced floral bulb formation, whereas a low cytokinin and GA ratio promotes vegetative bud in apple. Therefore, the limited efficacy of 6BA in mitigating GA3-induced flower abortion in this study may be due to the inadequate application rate of 6BA relative to the GA3. So, in conclusion, for high-trail variety like Navajo and Wachita, GA3 has several key features for commercial implementation. First, it has a favorable safety profile. Second, its application is inexpensive. Third, it has no negative effect on fruit quality. However, it's important to note that I think it is recommended to apply GA3 during the when the maximum chill accumulation is achieved and the risk of freeze damage is minimal. I would like to thank our Founty Souls and all my dissertation committee members and the Fancy Fund for providing experiment site. Thank you for your attention, and feel free to let me know if you have any questions.